Is this how life on Earth began? For decades, scientists searched space for the 5 standard DNA and RNA bases, A, G, C, T, U. They'd find 1 or 2 in carbonaceous meteorites, but always hit a wall. Once on Earth, it's hard to rule out contamination by terrestrial bacteria. To end this debate, Japan's Hayabusa 2 probe flew 300 million kilometers to the C-type asteroid Ryugu, digging up soil that never touched Earth's atmosphere, 100% space original. In the initial 2023 analysis, scientists only found uracil in a small sample. Was the universe's gotcha rate that low? Or did this asteroid just have E-tier luck? But in a new study published in Nature Astronomy, a team from Jamstack, Hokkaido University, and Kyushu University increased the sample size and finally hit the jackpot. The team got tiny rock fragments from Ryugu's first and second landing sites. To extract organics, they first used pure water, ultrasound, and cold extraction to preserve fragile molecules. Then they threw the residue into 6 molar hydrochloric acid, boiling it at 110 C for 12 hours to release molecules locked in minerals. Finally, this asteroid broth was fed into a high-performance liquid chromatography electrospray ionization high-resolution mass spectrometer to precisely measure each molecule's mass. The signal peaks for A, T, C, G, and U popped up one by one. That's right, the building blocks of your life's blueprint were all present, 300 million kilometers from Earth. But what if it was lab contamination? The team worried too. However, they found many isomers in the data, like 6-methyluracil, which are extremely rare in Earth's biosphere, unless a flush beats a full house. And combined with the fact that the extraterrestrial sample's base concentrations didn't follow Chargaff's rule, these bases were definitely created in space. It's the real deal. And there's more. The paper's biggest contribution is cracking the space kitchen recipe. Scientists compared Ryugu's data with NASA's Bennu asteroid samples, that 1969 Murchison meteorite from Australia and the 1864 Orgel meteorite from France. They focused on one metric, the Purin to pyrimidine ratio, or double to single ring ratio, since purines are complex double rings and pyrimidines are simple single rings. The Murchison meteorite showed an overwhelming number of double rings. Orgel and Bennu were overflowing with single rings. Ryugu, or star, was relatively balanced at about 1.1 to 1.2 to 1. Scientists found this wasn't simple. When they compared the double to single ring ratio of these rocks with the ammonia concentration in the samples, it drew a perfect negative correlation line. This means ammonia concentration is key. Imagine asteroids are chemical synthesis mobile games. They start with basic materials like carbon, hydrogen, oxygen containing malic acid, and nitrogen containing urea. If there's abundant ammonia at the start, the reaction smoothly forms simple single ring pyrimidines. If ammonia is extremely scarce, the system takes a hell mode hidden route, first becoming highly toxic hydrogen cyanide, then undergoing frantic self-polymerization to squeeze out complex double ring structures, leading to a massive surge in purines. This discovery strongly supports the hypothesis that when Earth was young, carbonaceous asteroids were mobile chemical factories, assembling simple atoms into life's building blocks and precisely airdropping them onto Earth. Amazing! It's insane. You know, platypuses have a beaver's body, a duck's bill, lay eggs, have venomous spurs, sense weak electric fields, and their fur glows eerily under UV light. They even have up to 10 sex chromosomes. It's a complete mess. It's almost as chaotic as a parliamentary debate, but a new study in biology letters, led by a multinational team from Ghent University, reveals its fur also hides a unique, hollow spherical melanosome, found nowhere else in vertebrates. Huh? Not that crazy? This is a textbook shredding discovery. Melanosomes are mini warehouses in cells that produce, store, and transport melanin. In mammals, whether round or flat, these warehouses are always solid. For 50 years, scientists thought, hollow. Melanosomes were exclusive to birds and always elongated or flat. That's because elongated hollow organelles, when strictly arranged in feathers, form nanogratings that interfere with light. The hollow air layer and outer keratin create an extreme refractive index contrast, producing dazzling structural colors. But when scientists built a mammalian melanosome database, they sectioned platypus back inside fur, put it under a transmission electron microscope and a scanning electron microscope, and found its fur cortex was packed with hollow melanosomes. Precise measurements showed these hollow organelles were about 319 nanometers long and 285 nanometers wide, with an aspect ratio of only 1.13, almost perfectly spherical. This is like forcibly merging mammalian and avian traits. Even crazier is its chemical composition. In mammals, elongated melanosomes usually contain eumelanin, producing black-brown, while spherical ones typically hold pheomelanin, producing red-yellow. Shape determining color is common knowledge in the field. But the platypus, to figure it out, the team threw hair from 10 platypus specimens into hydrochloric acid, heated it to 100 C for 45 minutes to dissolve outer proteins, then centrifuged, washed, added 100% ethanol, sonicated to fully disperse, and finally used a Fourier transform infrared spectrometer on the purified melanin powder to see what it was. Comparing the data to standard eumelanin from squid ink, they found these spheres primarily contained eumelanin, usually stored in elongated warehouses. This is why platypus fur appears dark brown. The platypus completely ignores the common knowledge that shape determines chemical composition. But what's it for? Scientists scoured databases of 120 other mammals. They even plucked and sectioned hair from platypus close relatives, long-beaked and short-beaked echidnas, and 6 Australian marsupials. None had this hollow structure. So, the conclusion is, it might really be for nothing. About 20% of their fur's warehouses are solid, the remaining 80% are hollow to varying degrees. Awkwardly, these hollow spheres are randomly scattered in the fur without the regular arrangement needed to form light-refracting arrays like in birds. So, while the platypus evolved a rare hollow structure, it can't produce dazzling structural colors. This is a biologically inexplicable redundancy. Scientists speculate this is a primitive trait left over from a common ancestor of ancient mammals and birds, or a vestigial adaptation for early aquatic life. The air trapped in hollow structures could offer minimal insulation, but since its relatives, the echidnas, later left the water, they lost this energy-consuming structure, leaving the platypus to keep it as a feature. Regardless, this discovery opens new doors for melon genetics and organelle evolution research. And thanks to the platypus, we have another ice-breaking topic for chatting with a crush. No need, because last time I shared about the platypus's two-headed penis, porcupines in Tainan? Yes, this isn't an urban legend. Fossil evidence just published in the Swiss Journal of Paleontology by Academia Sinica, filling a mysterious gap in East Asian animal migration history. But first, we need to understand the decades-long struggle of paleontologists. During the Ice Age, sea levels repeatedly dropped, turning the Taiwan Strait into a vast grassland connecting to mainland Asia. Giant beasts like mammoths, saber-toothed elephants, and rhinos roamed freely, but the migration paths of medium-sized mammals remained a mystery. Scientists found hundreds of porcupine fossil sites on the Asian mainland, but zero records on any East Asian islands, including Taiwan, Japan, Hainan, and the Philippines. We didn't know if these spiky creatures could cross the strait, or if they all perished halfway. Recently, a team from Academia Sinica noticed some unassuming teeth from the Chiding Formation in Zhuozhen, Tainan, including a well-preserved upper molar, a lower jaw fourth premolar, a molar fragment, and 3 broken incisors. Of course, you can't just call them porcupines. Seriously, you can't. However, as large rodents, their teeth have unique features like distinct high crowns and complex enamel folding patterns, allowing paleontologists to confirm at a glance that these belong to the genus Hystrix. But identifying the exact species is another matter. Researchers used electronic calipers with 0.01 millimeters precision to measure tooth length and width, and precisely calculated enamel height. Such detailed measurements are crucial because different porcupine species evolved specific tooth proportions and wear resistance to adapt to varying plant and bark hardness. They then fed the data into past software, creating scatter plots and using convex hulls for visualization. This is like drawing a strike zone for each known species on a chart. If the Tainan specimen's data fell into a specific box, they know its identity. First up, the small Hystrix lagrili with a total tooth row length of 21-22 millimeters. The Tainan fossil, estimated from a single tooth, had a total length over 35 millimeters, completely different, like a wild pitch over the batter's head. So, a large one? The Pleistocene of China had a giant species, Hystrix magna. Its strike zone was huge, but the Tainan specimen's data landed short, a pitch in the dirt, eliminating it again. Finally, the data perfectly curved like a masterful slider into the convex hull of Hystrix subcrustata. The umpire called it a strike. Who is it? It's the dominant species that thrived from southern China to Indochina during the Pleistocene. The fossil's geological layer dates back to the Middle Pleistocene, or 800,000 to 400,000 years ago, Taiwan's first porcupine record. Gotcha. This also marks the easternmost known distribution of porcupines, the first definitive proof of their presence on East Asian islands. Looking back, Zhuozhen, Tainan, 400,000 years ago was an incredible animal city. Besides saber-toothed cats, crocodiles, mammoths, rhinos, and giant pythons, there were even porcupines gnawing on tree bark. What else do you have, Tainan?